Chapter 8, Section 10, Biofilm Formation. Okay, 4 stages of biofilm formation that we've talked about before. Attachment, colonization, development, and then dispersal, because at some point in time that biofilm is going to become overgrown, depleting everything around it, and it needs to get to be else somewhere else. So when it comes to the attachment, this is just going to be random, okay? One random bacterial cell lands in a random place. It has a flagella or a pili that allows it to adhere to a surface, okay? Got to remember, 1 becomes 2, becomes 4. So if 1 can stick someplace, that 1's going to become 2. And if that first one can stick, there's a pretty good chance the second can stick. And if those 2 can stick, there's a pretty good chance the 4. And then the 8, the 16. Attachment is going to trigger what are known as biofilm-specific genes. This is an intracellular signaling and extracellular polysaccharides. Okay, so you get genes on the inside that are going to be altering metabolic activity, which is going to result in new extracellular polysaccharides beyond the NAG and NAM. A cell that attaches, that had flagella, that had pili, is going to lose those. Okay, it's no longer going to be motile. It's going to stay adhere. So the pili can stay, the fimbria can stay. Flagella, definitely gone. Once, say, the biofilm is established, or once it gets to a certain point that, certain quorum signals get too high, it may trigger the release of certain cells or certain number of cells from this biofilm, which will allow them to float away. They'll gain back their flagella, whatnot. They will, go through taxis to somewhere else where they may randomly attach, and then start this process all over again. So when you look at this, random cell comes into contact with the surface. The water is flowing through, flowing by. This water could be, a stream. This water could be a water pipe. This water could be the saliva in your mouth flowing over your teeth. It's all the same at the bacterial level. They adhere to something that normally they shouldn't. 1 sticks, 1 becomes 2, blah, blah, blah. They start to colonize. As they colonize and spread, get that gene expression that gets altered. Different metabolic pathways get turned on, others get turned off, and you start making polysaccharides that are going to lay down. Remember that meshwork. As it gets to a certain point, quorum signals, that signal protein, that signal molecule that they make and they excrete out, gets to a certain threshold level, triggers new capabilities. So instead of spreading out here, they start to go more vertical. And as they're laying down this polysaccharide, it truly becomes mesh-like, where you get these channels, so that instead of clumping, with the ones at the base of the clump are going to be deprived of nutrients, deprived of oxygen, whatever, by having these spaces in between, fluid can flow through, and with that fluid, nutrients come in, waste products get removed. But again, quorum signals are going to be such that it's going to hit a certain point where this just now becomes to a point of untenable growth. So certain cells are going to detach, gain back their ability to have a flagella, and they're going to swim away. So, bacterial binds. The binding is going to cause a transition from the planktonic, which is the grow of the swimmer, to, more of a sessile growing on and in a semi-solid matrix. It's a regulatory molecule, cyclic diguanosine. Okay, remember cyclic AMP? Well, here is a cyclic di-2-phosphate guanosine monophosphate version of it, C-di-GMP. This is the one and only time you're going to see this. Unlike the cyclic AMPs, which has multiple uses, the cyclic GMPs that have multiple uses, this is the one and only. Biofilm formation. It's widely distributed in bacteria, even ones that we normally don't associate with forming their biofilms themselves. When this accumulates inside of them, they have the ability to make this. When this accumulates, it's only going to be when they are associated with a biofilm that some other species has started. It's kind of cool. Synthesis and degradation, the build-up, the breakdown, this is all going to be based on environmental cues. Environmental cues that trigger certain cellular responses. This is the cyclic di-GMP. Cell signal tells two GTPs to be fused together. This right here, as this accumulates. Now, just the mere presence of, ooh, here's one. No, it's again, I keep hammering away at this in many, many lectures. It's not a switch. Yes, it's there. Start doing this. No, it's not. Don't do this. It's always thresholds. Low levels, cell ignores. As the level increases, certain things will start. When it hits this threshold, here, as it's going through that amount, that concentration, that threshold concentration, things get triggered. What gets triggered? New transcription factors. Riboswitches that we talked about in Chapter 7 will call for the expression of certain, or the translation of certain mRNAs that are under control. Proteins are going to be modified. So you're going to get the whatever it takes for the biofilm to be formed. And then at a certain point, other signals are going to tell phosphodiesterases to take this C-di-GMP, break it down into two individual GMPs. Remove the biofilm yes, yes, yes signal. Here's everything I just talked about from that figure in a written form. When you look at an example of it, Pseudomonas Originosa. Okay, this one forms what they call a tenacious biofilm because there's lots of polysaccharides there and other things from the environment that causes this to become really, really strong. By forming these biofilms, it also increases pathogenicity inside of us. They become impenetrable to antibiotics. So this is a classic opportunistic pathogen. Opportunistic means given the opportunity, it will hurt you. It will be hurt you, will do stuff to you. That's what a pathogen is. It's an organism, a microorganism that will do damage to a host. Opportunistic pathogen means that under normal conditions, Pseudomonas Originosa, it's there, it's on you, it's in you, it's not doing anything to you until something occurs. Now it switches and it becomes a pathogen that is actively trying to hurt you. That's what an opportunistic is. There's other ones, and we'll talk about those in later chapters. So we're going to see that normally it's in the soil. So you're coming in contact with dirt, dust all the time. You're coming in contact with this all the time. Under normal conditions, when you're healthy and everything's hunky-dory, you're not getting sick. But for some reason, this day at this time when you came during that exposure, your immune system dipped, or you had a wound, or something going on. Next thing you know, it's in your blood, your lungs, urinary tract, ears, skin, somewhere else. Turns out Pseudomonas Originosa is a big problem for cystic fibrosis patients. Okay, because it forms these big, thick biofilms down in the alveoli. Remember, a cystic fibrosis patient, they're already having trouble thinning their mucus because they don't have that transport protein that pumps out the ions that will thin out the mucus, which for us, people who are wild type and have it and are expressing it, every time you breathe out, mucus is coming up into your throat and you're swallowing it. That's the norm. Cystic fibrosis patients, their mucus never thins. It stays really, really thick. And it means it's not coming up whenever they breathe out, so it accumulates. Well, Pseudomonas Originosa gets down in there, gets into that nice nutrient-filled mucus, starts growing, starts to form these biofilms that literally will wall off certain alveoli sacs or certain bronchioles. So becomes an issue for them. Quorum sensing, remember this, as I said already in this lecture, quorum sensing. It's a single molecule made by one bacterial cell that is excreted out that, auto-regulates self, comes back on self, auto-signals, but then can disperse out to other bacteria in that microenvironment, triggering responses in them. Acyl homoserine lactones, AHLs. This is one of the big quorum signals. As this accumulates in the bacterial cell itself, because remember it makes it, excretes it out, it may come back in, or it may disperse in the the fluid of that microenvironment and other cells pull it in. So as it accumulates, it triggers the making of the C-di-GMP. Low levels, the cell ignores it. As the levels creep up, more and more things start to be triggered until it hits a certain level, and then complete change of metabolism, change over the metabolism, change over which genes are being expressed. What's interesting is cells that get damaged because of toxins, because one of the things that the AHLs will also do is they will trigger toxin production, kind of similar to what the endospore formation, remember those that were rushing to do their endospores from, what was that, section 7? Well, toxin just like that can be released by certain cells as the AHL levels increase, which will trigger the lysis of competing cells and the release of that big circular chromosome, which will entangle and hold other proteins and stuff right there. And it will entangle into that polysaccharide, layers, and just add in more structural composition. It'll help promote the formation and laying down of more polysaccharide. Yeah, so using the bodies of, or the cell debris of the, the dead and the dying to build up this, covering for the biofilm, this protective covering. Yeah, can't see anything in that picture. I can sit there and go. So we're just going to skip it. We've been talking about the past couple of slides, Pseudomonas originosa. Its genetic cousin, Pseudomonas Fluorescence, uses a slightly different strategy, and I am not going to go through and it's, instead of the CD, cyclic, what was it, GMP here, it's using a different version of, and I'm not going to go through all of this. You can read on your own. Vibrio Cholera, another one, uses biofilms with polysaccharides and carbohydrate binding proteins. RBMA, RBMC, BAP1. It's grabbing carbohydrates, pulling them in, holding them there to not only stabilize the biofilm, add in more protective layers, but those carbohydrates can at a later time point become a fuel source. When you look at the Vibrio Cholera, you can see that as it's growing in, this is increasing, this biofilm is increasing in mass. Certain genes will become expressed, other ones will be turned off, depending upon size and complexity. What we see here with Vibrio Cholera is that at low density, low numbers, biofilm formation as a protective mechanism. But when the biofilm gets bigger and bigger and you now pass a certain threshold, and I keep using that term a lot because get used to it, a certain number, the density of the number of bacteria present, well, now it's going to go the opposite way. Start breaking apart, start spreading elsewhere, getting too big to maintain. Whereas Pseudomonas Originosa, it was low levels, low biofilm expression genes. As those numbers increased, even more expression or new biofilm genes and operons get expressed. So they're going in opposite directions here and how they are reacting. I really don't want to talk about Archaea.